Then soil samples are collected using soil auger at the depth of 0 to 15 centimeter. Global position system were used to make the soil sampling position points. For each soil sample, we take data sheets were used to each and every soil sample. In this data sheet, we collected some information of that location: DS division, ASC region, GN division, name of the year, longitude and latitude, sample number, irrigation method, and if there are any special problem, it also mentioned in this data sheet. Then collected soil samples analyzed at two research stations at RRDI and FCRDI, and these samples analyzed using standard chemical analysis methods. And soil sample were analyzed for available phosphorus, exchangeable potassium, exchangeable zinc, soil pH, electrical conductivity, and soil organic matter. Then collected information were used to prepare the maps. For preparing for maps, use ArcGIS 10.5 in the Geographic Information System. Then the data were grouped based on soil test values reported by the Soil Test Based Fertilizer Recommendation 1997 and 2013. This legend gives the categories of different parameters. Then this table shows percentage land extent of rice growing soil in paddy fields of the Polonnaruwa districts for different soil parameters. Then this spatial variation of soil available phosphorus, more than the available phosphorus level of this area, consider the more than about 58% of the land extent is higher than the 10 ppm. So soil phosphorus values varied from 1 to 53 milligram per kilogram, and showed the average as 8.9 milligram per kilogram. This variation may be due to difference in absorption and desorption mechanism of phosphorus in paddy soil, and level of phosphorus application in the previous season, or due to level of ferrous and calcium in these soils. As the DOA recommendation, when the soil available soil phosphorus level is more than 10 ppm, we need not apply any phosphorus containing fertilizer for that rice fields under irrigated or rain-fed situation. Then the spatial variation of exchangeable soil potassium, when we see this table, we see the 100% of the land extent is less than 160 ppm phosphorus. When the soil available soil available potassium level is 160 ppm, we have to apply potassium fertilizer for that soil. The average value of the soil exchangeable potassium is 53 and maximum value is 133. The whole district's potassium fertilizer should be applied as recommended, because there is no place having excess potassium. This is a recommendation for potassium fertilizer application for irrigated paddy. When the exchangeable potassium level is greater than 160, we need not apply potassium fertilizer, but this area, all the location are lower than the 160 ppm. Then exchangeable zinc level more than 82% of the total paddy area has a very low level of zinc. And your recommendation for zinc is 5 kilograms per hectare. Therefore, we have to apply more zinc sulfate is applied to many locations for this districts. The soil pH is 67% land extent is in between 5.5 to 6.5, and the average soil pH was 5.5. The optimum soil pH for rice cultivation is around 5.5 to 6.5. So 67% of paddy fields have optimum pH value. Then electrical conductivity, 92% of land extent is less than 0.15 decisiemens of meter value, and then average EC is 0.1 decisiemens per meter. Maximum EC value for the district is 0.9, and EC is less than when the EC is less than 0.15, no soil salinity is there. Therefore, 92% of the land extent is not subjected to soil salinity in this district. Then spatial variation of soil organic matter. Only 2% of land extent having more than 3% organic matter. Then soil organic matter in the districts is very low, showing the average as 2%. 98% of the paddy fields are very poor in organic matter content. For an optimum crop production, organic matter content should be more than 3%. Using this information, we prepared maps. These maps are included in Google Earth Pro. If you want to use this map, you can open Google Earth Pro and open the file extension.kml. Using this map, you can take the information, fertility information from that map. This is the map in Google Earth Pro. When you open the Google Earth Pro, you will see this map, and you can find your need point, and you can get the information for this map. Conclusions are 58% of land extent have excess available phosphorus. Soil exchangeable potassium is low in 98% of land area. Soil salinity is not critical in 98% of paddy fields in the district. 82% of land area show critical level of zinc, and very low level of organic matter in 98% of land extent. The land parcels where remedial measures should be applied can be easily identified through the prepared maps even without analyzing soil samples in each field in all the time. The authors wish to acknowledge Sri Lanka CARB for providing financial assistance under National Agriculture Research Project to conduct this project, and all the field officers who assist for soil sample collection in the Polonnaruwa district. Thank you.
Thank you. Any questions, the comments? Yes.
I would like to ask one question. This is very good research. I want to know that you have get information from the farmers whether they have cultivate the both Yala and Maha both Yala and Maha season paddy cultivation. Otherwise, sometimes the farmers use more phosphorus fertilizers for the vegetable cultivation. In that case, if you come to an conclusion that all the fatty fields are, that means phosphorus is high in fatty fields, sometimes this is due to the cultivation of other field crops and vegetable as well as those things. So paddy by cultivation paddy is not, I think, the only reason for applied more phosphorus fertilizer.
No, this is Polonnaruwa district. These all locations are both season cultivate rice.
Yes. Now, this is important because you have collected data, this organic matter data, right? Less than 3%. So I heard you may be knowing Dr. James, right? Handawal, soil scientist. So he said 2% of organic carbon, I suppose, I can't remember that it's organic matter, organic carbon, will not be found in Singaraja forest also, right? So organic matter content may be very low in our soils. Now, with the introduction of this boothia, the harvesting machine, the straw will be chopped and added into the soil, right? So, you have any idea of, using harvester in these areas? Number one. Number two, it's related to previous question, right? Whether they are cultivating both seasons paddy, or there may be places where they cultivate only one season paddy.
Yeah.
So,
but this is only rice cultivation, and the other thing is some fields they use boothia, but they may apply rice straw in the field, but in the data sheet some information we got the some organic matter application or anything, but that is not include this one. Maybe some location may the increment of the some organic matter increment due to this
use of this harvesting machine. Okay, because when I was there at RRDA, right, when our minister was, the previous minister, I can't remember his name right now, but anyway, Dimu Jayaratna. So he said, by applying organic matter, you can control even weeds, right? So I was just thinking whether people have been applying organic matter into the paddy fields, because that was one of the recommendations, Dr. Amarthy wanted to do, to make use of, organic matter straw in the field. Yes. So if that is happening, so better to collect that sort of information also while you are doing this, right? So that you will get some very good idea.
Yes, sir.
So that's only a suggestion. Yes. Thank you very much.
Thank you, sir.
Yeah, can you have the mic over there, please?
Right, I am extension officer. So when the phosphorus content in the soil is increased, normally we know there is the antagonistic effect is there.
Yeah.
So when you consider about zinc, you concerned about, or you mentioned about the zinc. The other thing is calcium. So did you concerned, or did you have given any attention about calcium uptake? Or we know that there is an antagonistic effect, no? When
Yeah.
the soil phosphorus is increased.
Yeah, in this.
The other thing is, when you consider about the soil in the dry zone, the manganese, the micronutrient manganese, is also in the very minute level with regarding the phosphorus. So have you
But
considered?
in dry zone, where the manganese and calcium, in rice fields, there is no problem in calcium and magnesium. There is enough calcium and magnesium in dry zone. But in wet zone, there are some problems. Another thing is the antagonistic effect is also some in dry zone, some situation is there. But that's why we prepare like this maps. When we using this information, we can reduce unnecessary application of fertilizer. We know the phosphorus and potassium, especially phosphorus level in that location. We can cut down the phosphorus level in next season.
Yeah, Doctor.
Now, Ms.
Nimar.
In one of your conclusion, I saw that 62 or 67% of the area is already have enough phosphorus. Is it correct?
Yes, yeah.
Okay. Now, when was this study done? Which year this study was done? When was
This sample collection was done in 2017.
2017.
Yeah.
2017. Now, my question is, now, are the farmers use phosphorus fertilizer in those areas? 67% is a huge acreage. Are the farmers using phosphorus fertilizer for those areas right now? That's my question.
Yeah, some farmers use, but this technology is given to that area. This Polonnaruwa district, the after develop this technology, all the information given to this district, Polonnaruwa district, and some are using this information. If they use this information, they can cut down the phosphorus level. And however, some farmers use phosphorus fertilizer for their paddy fields.
Yeah, it's a problem because it is it's very expensive
Yeah.
import phosphorus, and then we are doing kind of pollution, environmental pollution, and then when the information is there, and why don't we produce, forward it to the authorities, and then take the necessary action immediately, explaining the situation, and then why don't we take that message up to the authorities? Anyone in the audience from the extension division, senior officers from the department, so that we can follow Naru for that matter from the head office?
Yeah, this question is for a person from the extension division. Why this has not gone to the farmers, and why are they still applying more fertilizer when 67% of the land has more phosphorus? So that's the question from the former director.
I think another author is there.
Ms. Ratnayake is there?
No, Mr. Sirisena is there.
He's here?
Yeah.
Yeah,
He
he's
has some experience.
behind you.
Yeah. Right, please.
No, what I can say that fertilizer is given on subsidy rate, on subsidy at the rate of we say 50, I think 5 Today it is given
1,500
1,500 rupees
per
for
50
50 kilos. So that you can't keep that in your house because it is belongs to the government. So that farmers are compelled to apply it. So then what we will have to do, what we did was, we went to the extension office and we educated them, please use this information when you recommending phosphorus fertilizer. I think as Dr. Disanayaka mentioned, it is the duty of extension officers to educate farmers and to ask them not to apply, keep that fertilizer and apply it for some other vegetable or some other place. That is the only option which what we can do.
Potassium, zinc and other elements are lacking in those areas. So instead of phosphorus, they can apply, they can be given more potassium fertilizer.
Yeah.
Or more nitrogen if it is case. But this has to come from the fertilizer secretariat and from the ministry. So recommendation has to be, for specific areas with reduction of phosphorus for that area.
Now you are still a young officer in that, you are from RRDI.
No, sir. Now I am in fruit research and development.
Well, whatever it is, you can push that message to the higher level and let them decide, right? We can't force you to do that, right? So you can give the data and this is the situation. And Dr. Sirisen is there to back you up. So that is the only way where you have to give this message, immediately to the extension.
Thank you, sir.
Thank you. I think your presentation made a lot of interesting discussion, and perhaps we could continue, and ultimately that might go into the policies and national politics also. Right, so with that, thank you very much for your interesting presentation.
Thank you, sir.
Right, now we have come to the end of the session, and I am very pleased to note that this young scientist from the Department of Agriculture is aligning their research in order to address the challenges, especially in soil and water sector. So these presentations were basically with regard to soil fertility and climate change, and these are the topics that are currently in discussion. And also I'm very happy to note that they are using the latest technology and analytical tools, unlike in the past, for their research work. So thank you very much for your presentations and interesting papers. And next I would like to thank the audience for their lively discussion and participation. And finally, I would like to thank the organizers for inviting me to chair this session. And thank you all very much. And then I'll hand it over to Shiromi for any other announcements. Thank you.
Thank you, sir. I would like to thank to Professor Gunawardena for his marvelous job as a chairperson and to the panel of judges for their excellent work. Also, I must thank for the invited presenter, Mr. Kapila Munasinghe, and all other presenters, and also all who have actively participated for the discussions and giving their comments, as well as to the audience to encouraging the presenters by presence here. Before winding up the technical session 3, Soil and Atmosphere, it is my duty to invite to present the token of appreciation to chairperson and the panel of judges in the session to appreciate their excellent work in this session. So, I cordially invite Dr. Ranjit Punyawardena, Director in RMC, to present the token of appreciation for Professor Gunawardena. Sir, please. Also, I kindly invite sir, to present the token of appreciations for the panel of judges. Dr. Mrs. Varshid Andeniya, Dr. Mrs. Padmini Girihagama, and Mr. K.M.A. Kandaragama. Please come forward. Thank you very much, sir. With that, I would like to wind up the technical session 3, Soil and Atmosphere. There are a few more important messages for you. The morning tea is ready at the outside lobby. The organizing committee of ASDA 2019 are invited to enjoy the tea. The next technical session will be started at 10:30 a.m. All are invited to present for the next session as well. Thank you.
thesis supervisor in the University of Colombo and also as a senior consultant to the Faculty of Urban and Aquatic Bioresources of the University of Sri Javadanapura. So then I would like to introduce the panel of judges. Dr. Mangalika Nugaliyadda, former DDR of the research station at Sita Elia, and also Dr. Professor Kapila Yakandavala, who is a professor of Wayamba University of Sri Lanka, and also Dr. Shanika Jayasekara, senior scientist of the Sri Lanka CARP. So, Dr. Disanayaka, sir, I invite you to proceed the session number 5. Thank you, sir. So, we will wait. Only 2 judges. Yeah, thank you.
Good morning to all of you. So therefore, everyone gets 20 minutes, and out of 20 minutes, as in the previous session, 15 minutes for the presentation and 5 minutes for the questions and discussion. Therefore, please adhere to the time given for your presentation. So the first presentation, invited presentation, is by Dr. Jinadri Soisa. I need to give her a little introduction because she retired about maybe, I think, 7-8 years ago from the department. Those young people may not know her, and so therefore I need to give her a little introduction. Dr. Jinadri Soisa graduated from the Faculty of Agriculture from University of Peramia in 1975, and then she joined the Department of Agriculture as a probationary research officer and as a the confirmed officer in 1976 as a plant pathology to work on plant pathology. She obtained her PhD degree from Imperial College London, again on plant pathology in 1983, and she continued to she worked at Mahalippuram for 7 years. And then she came to Gannur again, and she was the plant pathologist. Unlike nowadays, there was there are few positions like for the department, plant pathologist, chemist, entomologist, like. So she was the plant pathologist, and she was the deputy director of research at HODI, and then director. Finally, she became the Director General of Agriculture in 2009 to 2010. She was the first woman to hold the position, this prestigious position in the Department of Agriculture. With this little introduction, I invite Dr. Jinadri Soisa to make a presentation. Thank you.
Introduction, and I also thank the organizers of ASDA for inviting me for this presentation. Actually, when Dr. Lakmini invited me over the telephone, I was a bit hesitant to accept it because I am not very conversant with the inside developments of the Department of Agriculture recently, and I contacted some eminent scientists of the Department of Agriculture, and they have given me some suggestions and comments. So I thank them also before I make this presentation. So in line with your theme, ASDA theme, I have selected this topic, plant pathology research for safer tomorrow. The introduction is reasons for safer research, health hazards and environmental pollution caused by excessive use and misuse of agrochemicals. You all know that. And considerable changes in people's attitudes towards the use of pesticides in agriculture, and organic crops fetch high prices. So there is a need to restrict toxic chemical pesticide use, and there is political pressure to remove most hazardous chemicals from the market. Consequently, researchers are trying safer alternatives. So in pathology research, we have two broad categories: disease epidemiology and disease management. In research, in our department, I have attended some working group meetings recently, and I have seen disease diagnosis and therapeutic measures of disease management were given more attention than the other topics. So I will talk about this disease diagnosis, research and therapeutic measures of disease management research in this presentation. So in disease diagnosis, correct disease diagnosis is very important for effective disease management. Our researchers commonly use now these serological methods and molecular methods because they are quick, efficient, and sensitive. But misidentification may result in with serological and molecular methods if, because they are very sensitive tests, so you have to be very careful. Presence of inhibitors in plant sap, positive controls are not available. If chemicals are used not up to the desired grade or not properly stored, antisera, if the antisera are not specific in ELISA, related pathogens give positive results. So in molecular tests, molecular markers, if the molecular markers are not specific, amplification of non-target regions may occur, and purity, if the purity of nucleic acids is low, that is also will give misidentification. So you have to be very careful and verify pathogen identification by conventional methods. So you can confirm the identification of the pathogen with these methods, but not possible to confirm the disease unless you perform Cox postulates. All of you know what Cox postulates is. So I will talk about disease management. There are several methods. First one is cultural, second physical, crop resistance, biological, chemical measures. All these are somewhat safe. In chemical, there are toxic chemicals, and many fungicides we are using have chronic toxicities. Their acute toxicity is somewhat low, but they have chronic toxic substances. So we will go on to the crop resistance first, and plants are evolved to have their own defense system against pests and diseases. So use of resistant varieties is the best method of disease management, as safest, least expensive or no cost to the farmer, easiest, most effective, and compatible with other management measures. So it is better officers should give the high priority for disease development of resistant varieties for safer agriculture. They should be given high priority. And first one is conventional breeding, collaborate with plant breeders and carry out varietal screening. And mutation breeding also with them. There is very good teamwork in rice and development of rice varieties. So likewise, the plant pathologists should give high priority for development of resistant varieties, mutation breeding, use of variants of tissue culture. Disease resistant could be selected from regenerated variants or plants, because in tissue culture we are using some mutagenic chemicals, so mutants will occur. So it is better, in some countries they have selected these variants and disease free and disease resistant varieties were developed. Tissue culture disease free in potato, strawberry, banana, sugarcane and cassava. In other countries they have selected genes resistant to diseases. And cross protection, it is something like vaccination. Prior infection with a deactivated virus prevents or interferes with subsequent infection by another isolate of the same virus or closely related virus. So there are quite a lot of work done in other countries, and it is like vaccination of human beings and animals. You all know about the gene transformation, so it is better you focus on development of rice varieties, not only rice varieties, other varieties also for safer agriculture. Then I will come on to research on biological control. In biocontrol, researchers are manipulating antagonistic microorganisms or biocontrol agents found in nature. So we have direct antagonism and indirect antagonism. In direct antagonism, the pathogen is directly attacked by a specific BCA that kills it or its propagules. In indirect antagonism, results from activities that do not involve sensing or targeting a pathogen by BCAs. It has competition, induction of host resistance, production of antimicrobial substances, and antibiotic mediated suppression, and lytic enzymes and other byproducts. In direct antagonism, there are obligate bacterial pathogens, hypovirulent strains, facultative parasites, microbial predators. There are quite a lot of examples in other countries, and these are all commercialized products. In indirect antagonism, competition for nutrients or space, the consumption of exudates or leachates, consider for scavenging, prevention of iron absorption, physical niche occupation, and also some microbes induce defense pathways, stimulation past host, plant host defense pathways. Some biocontrol strains of Pseudomonas species and Trichoderma species are known to strongly induce plant defenses. This is Trichoderma. And production of antimicrobial substances, you all know some microbes produce antibiotics. So Pseudomonas fluorescens produce DAPG antibiotic for controlling, and this is used to control damping off of the Pythium species. And crown gall caused by Agrobacterium tumefaciens was used controlled by Agrobacterium radiobacter because it produces agrocin 84. Likewise, there are quite a lot of microbes producing antibiotics are used as biological control agents in other countries. Production of antimicrobial substances, lytic enzymes and other byproducts of microbes. Lytic enzymes, chitinase, glucanase, glucanase, protease, lipase, those things are produced by microbes. You know, the
very commonly used Trichoderma harzianum, produce glucanase, chitinase, protease, and lipase. So they induce resistance, and also they coil. This is pathogen, and this is Trichoderma. So it coils and causing lysis. This is Trichoderma. This is very rarely found in nature. Byproducts of lytic enzymes, oligosaccharides, chitosan. This is a commercial product. Chitosan is a non-toxic biodegradable polymer produced from chitin. Treatment with chitosan increases resistance to pathogens. And the most effective biological control agents appear to antagonize pathogens using multiple mechanisms. Examples, a lot of pseudomonads produce DAPG, induce host defenses, and aggressively colonize roots and contribute to their ability to suppress pathogen activity in the rhizosphere of plants through competition for nutrients. And for successful biological control research, our researchers use this, many researchers report or publish data here. They select biological control agents from lab experiments, and they report and stop at that stage. If it does not reach the farmer, it's waste of time, effort, money, and other resources. So it is better to work further, work on mass production of BCS, storage requirements, nutrient requirements, antagonistic mechanisms, product formulation, etc. And test in research fields, provide all necessary for disease development and make sure that disease occur at high level in the field. Provide different strains of the pathogen, use a mixture of BCA if possible. Coordinated trials at different locations should be carried out and then test in farmers' fields and commercialize. If you not do all the things and commercialize, your time is wasted, effort, money, and other resources are wasted. There is a biological control, successful biological control, safer method in the lab, but if it doesn't go to the farmer, it is really unfortunate. Commercialization of biological control agents. There are some examples of successful biological control agents of plant pathogens tested in farmers' fields by our scientists, and farmers' acceptance for those had been very high. Those should be commercialized forthwith to replace toxic fungicides. Here we are using toxic fungicide, we are recommending toxic fungicides, keeping our safer biological control agents in the lab. In other countries, many biological controls are used as commercial. Are some of the products from India. So we also should go for that. And storage or preservation facilities for biological control agents. This is a responsibility of the directors, heads of divisions, and scientists. Scientists have used public funds and resources to develop biological control agents. Before commercialization procedure, effective biological control agents should be stored properly, as they are a valuable property of DOA and the country. They should be stored in respective research institutes, and a reference or duplicate collection should be maintained in a central place, because otherwise those will be lost forever after retirement or leaving of the respective scientists. Make those available to fellow scientists purpose, giving due recognition or royalty to the founders. So it is better, we should have facilities to preserve or storage biological control agents found so far. Regulation and quality control. Regulation for use and commercialization of biopesticides and botanicals had been formulated a few years back. Unfortunately, procedural obstacles are preventing registration and commercialization of promising BCAs. This deprives safer methods to farm environment and, above all, safer products to consumers, making all of us losses. Relevant scientists, directors, ROP office, private companies should work out a simple procedure for commercialization, as we should contribute to non-toxic agriculture in a big way. Scientists should work with relevant companies for maintaining quality of BCAs regularly after commercialization. This is actually I heard that some companies are willing to market biological control agents found by our scientists, but still there is something delay in this process. Then we will go on to the safer chemical management, that is induced resistance. Now there are compounds generally regarded as safe. Call it grass compounds. Examples are salicylic acid, jasmonic acid, dichloroisonicotinic acid, arachidonic acid, ethylene, xylase, cysteamine, chitosan, and PR proteins, that is pathogenesis-related proteins, glucanases, chitinases, lysozymes, peroxidases. Those things are, most of these things are now commercially available. So we can use these things, test these compounds, and recommend this for the farmers. These compounds act as elicitors for induced resistance in plants. Induced systemic resistance signaling by jasmonic acid and ethylene, systemic acquired resistance signaling by salicylic acid and PR proteins. So we test these things, and some of our officers have tested chitosan. I know that Dr. Rajapaksha has tested chitosan and found it is very effective. In post-harvest disease control, treatment with chitosan stimulated microbial degradation, amendment of plant growth substrate with chitosan suppressed root rot caused by fusarium in tomato, treatment with chitosan suppressed anthracnose in capsicum found by Dr. Rajapaksha. This should be recommended for disease control forthwith. And these grass compounds, they show highly antifungal and antimicrobial activities, inhibit spore release or germination, strengthen host cell walls, break down pathogen cell walls. These chemicals could be applied to plants by injection, foliar spray, or seed treatment, post-harvest treatment, soil drench, etc. Mini grass commercially available. Research on this area should be strengthened and pass the mature technologies to farmers quickly for safer tomorrow. If we delay these processes, actually we are failed in safer agriculture. So acknowledgements, I acknowledge very much the comments and suggestions of the following scientists: Dr. R.G.S. Rajapaksha, Dr. Tushara Wickramarachchi, Dr. Jayant Senanayaka, Dr. Lionel Nugaliyadde, Dr. Manganika Nugaliyadde, and for coordination, Dr. Lakshmini Priyanta. Thank you.
Thank you, Madam, for the very excellent presentation. Now the presentation is open for comments, questions, and discussion. Please identify yourself so that others know who you are.
I am in the Department of Agriculture and retired and working again in PQS Katunayaka, Dr. Soisan, she's actually one question. You mentioned about the biological control.
Yes.
And I want to ask how long it will take to complete the research for commercialization steps, the identification and up to commercialization stage, how long it will take?
It is actually depending on the biological control agent and also the researcher. If you speed up this process, you can go for the field, for the lab trials, lab experiments, it don't take much time. But you have to work on mass production, storage, and lot of other things. It's a long time. And for field trials, at least 2 or 3 years it will take. So it is better. Now I think Dr. Rajpaksha has done quite a lot on it. Dr. Rajpaksha can tell about this, how long he has taken it to a commercialized product.
Yeah, actually, madam, it take at least 4 or 5 years, because actually the initial step we can do within 1 year, but later we need to go for the field trials in different location, in different season, like, and we need from the bioefficacy at least nearly 5 years.
Yeah.
5 years.
And if you do coordinated trials with several researchers, you can cut down the time period. Because in different locations you have to test, no? If in one season you can test with several, like in coordinated fungicide trials we have done in the past, so you can cut the time. Still, some researchers have found them, so why don't we take on so long and commercialize? So it is up to the to take this thing out and speed up the commercialization procedure.
Any other comment? Yes, sir.
Yes, I am Ranjit Mapa from National Research Council. Now, after successful biological control is found and recommended, what is the path for the commercialization? Is there any kind of way that the agriculture department can, yes?
Yeah, now it is by the registrar of pesticides, I suppose, Rajpaksha. He needs some information about this thing, and there should be a person or company to market this product. And Dr. Rajpaksha has found some biological control agents, I understood, and he says, are asking it, their acceptance is very high, and there is a company willing to market it. But our procedure in the ROP office and other places, so it hinders this. There are some obstacles. So somebody is here in this, working in the process, ROP office or somewhere, why it is not going into the market, actually you can tell.
As a follow-up question, I like to know of any examples that these things have been now commercially available. Is there any
in other countries, quite a lot available. In India, I have shown you some products, but we can't bring those things because those are microbes.
Locally, are there
Locally, Rajpaksha has developed, and when I was there, I have developed some, and we have green farms, and they produced it. They have formulated their own product and used it for the thing. We couldn't go beyond that. The problems are there. So anyway, Rajpaksha can tell what happened.
That actually there are guidelines for the commercialization of biopesticide or plant products, but that procedure actually, this procedure controlled by the pesticide register. It's a guideline and procedure, but there are some problems to commercialization of this.
So, I want to add something. As I visited the Asian cutting that nursery exporters, they are making their own trichoderma preparations. And
is it green farms?
Yeah, Asian cutting.
Yeah. We have
so
given.
I think they will gradually catch up.
Yeah. For it is we can give the whole community, farming community, if somebody is marketing it. Department can't do marketing, no. So it is better, and if the companies are willing and farmers are asking, why don't we give it? So that's.
Any other comments? In the absence of any other comments and questions, I'd like to thank Dr. Jinadri Soisa for excellent presentation. And also I'd like to add that the last week there was a conference on the association by plant pathology and microbiology science faculty, where the lot of members working from Columbia University, departments botany, department of pathology, department plant sciences, Jawardhanapura, Peradeniya. So what I am going to say, in isolation there are a lot of work being done, but it is very unfortunate that they don't collaborate, they don't work together. Every time that we must get, collaborate with other, the researchers in the, especially university system, and other system like tea research, rubber research, there are officers working in specific areas individually. I think it is high time that we must get together. Pathogens are common, so therefore, the lot if we work together in collaboration. With this comment, yes, madam.
And I have to say that now, if we can store these biological control agents in a central place, we can request those scientists also to give their biological storage and keep it for as a sort of a property for the country, of the country, property of the country.
Yeah. Actually, my observation attending that conference was that they are doing a lot of preliminary work which are kind of very positive in vitro test.
Yeah, that's.
But we need to take it to the field and see whether it works in the field. So, that is where we can get together. So everybody gets credit, and finally the country is benefited at large. So, yes, ma'am. So thank you very much. So as an appreciation of a good presentation, we will give a round of applause. And there's an announcement by the.
So thank you, Madam, for your very informative presentation. In this moment, I would like to invite Dr. Shiromani Rajapaksha, Director of the Fruit Research Institute, to hand over the token of appreciation to Dr. Jinadri Soisa.
With this, we are now to begin this technical presentation. The title of the technical first presentation is Occurrence, Taxonomic Identification and Life Cycle of Fall Armyworm, Sadoptera frugiperda in Sri Lanka, presented by MTMDR.
Morning, Chairman, sir, distinguished scientists, panel of judges, and dear colleagues. To present occurrence, taxonomic identification and life cycle of fall armyworm in Sri Lanka. It was invaded to Sri Lanka without proper approval from our border agencies. Sphodoptera frugifoda Frugiperda belongs to the family Noctuidae of the order Lepidoptera. And Frugiperda means in Latin for lost fruit, named because of the species top. It is native to tropical and subtropical regions of the America and first identified in Georgia and named Caradrina frugiperda in 1797. After that, invaded the African continent to be spread across 44 countries in sub-Saharan Africa. After that, in 2018, it spread to India at Karnataka state and then to Thailand, and in 2019 to Bangladesh, China, and Myanmar IPPC records. And this is the global distribution of fall armyworm. At this moment, it is in these areas, as maybe after that it will go to other part of the world. And the larvae damage crops. It's a voracious feeder and feeding more than 80 plant species and prefers maize, but also rice, sorghum, cotton, and some vegetables. Moth can fly up to 100 km per night with wind, and female lay up to 1,000 times. The egg cluster has about 100 eggs. Crops affected in 3 ways: plant stand reduction, direct damage, leaf defoliation. Larval densities are usually reduced due to cannibalistic behavior. This is the cannibalistic behavior seen in our. And also they are as similar to other Lepidopteran, it has an adult male, female, egg, and after that it will lay eggs, larvae, and pupae. And let me first describe about the first occurrence in Sri Lanka, fall armyworm. It was in 18, Assistant Director of Agriculture Mahawaya phoned to Plant Protection Service Gannuruwa and informed about an unknown pest. And actually he little know about this because during our agricultural promotion week, we have included this leaflet. According to that, I went to Idapola Viltalava maize field and visited the farm of Mr. Siripala. And the variety was specific 987. We collected samples and reared at plant quarantine unit of Gannuruwa. And also morphological identification was done using the literature available in the internet, and reported to the Director General of Agriculture on 30th October 2018. And this is the objective of this study: morphological identification of fall armyworm, taxonomic confirmation, and biology and life cycle studies in Sri Lankan conditions. And also, I will describe the methodology. Samples were collected, and maize leaves were given for larvae and replaced every other day. Adults were fed with 10% solution of bee honey, and 6 insta larvae and adults were subjected to taxonomic identification. Use this tell you about this. And also regarding the life cycle and biology, larvae were reared using circular insect breeding dishes containing maize leaf pieces and maintained at 28 plus or minus 2 degrees centigrade and 75 to 80% RH. Larval head weight removed after each egg diasis and measured the width of head capsule using an ocular micrometer at the widest point of the vertex. Adults were reared in plastic oviposition cages to obtain eggs. It was inside another insect to allow larvae to escape from the lab. Paper towels were lined as oviposition substrate. The eggs were examined at an interval of 12 hours till hatching. And also, I think all of you also know armyworm damage male crops in different growth stages. During the foliage stage and also to the whirl, this is also to the whirl, and also when cobs are forming it will eat some silk in the cob, and also after the seeds. And I will describe the specific characteristics of larva. As given in taxonomic is, it has an inverted Y shape in the head area. And also there are two paler lines in the whole body. This is the literature and this is our specimens. And also at the 8th abdominal segment, it has 4 distinct spots in a square. These are our specimen photographs. And also, this is actually aligned with the literature, and because I have the literature, and also this is the polar sample characters. Inverted Y's in head capsule was present in both, and dorsal spots on second last body segment, that is 4, and also second last body segment is the 8th segment, that is both present. And broad pale band along top of the body and dark side is present. 6, skin smooth under magnification of 25 power without microspines. Normally, other Spodoptera species have microspines, but this doesn't have microspines. Young larvae greenish, yellowish, and 6 to 9 centimeter in length literature, but our got 5 to 9 centimeter length. Mature larvae brownish, 30 to 36 centimeter. Our sample was brownish, 29 to 35 centimeter long. And also, let me explain the taxonomic characteristics of forwing of adult male. This is very distinguished characters. Actually, you can see that number 1, that is rainiform spot, partially outlined in black with a small sideways V-shaped marking. This is the V-shaped marking. It is present in our Idopolis sample and also maybe in other Spodoptera adult samples at the moment. And the second one is orbicular cell, oblique, and not as narrow as in the species with strongly contrasting transverse lines, because others have been contrasting transverse lines. And also third one is row of small black cover glass-shaped markings near apical margin of the wing. This is the main characters of the literature. And also these characters are similar in, this is the adult male. Adult male has these characters, but I will explain later about the adult female. That is, upper half of the wing is this wing in dark gray and brown, and over half to post-medial line or beyond pale gray or brown. An oblique white bar in anterior apical corner. Orbicular spot light brown, that was I explained earlier. And ventral is dark gray. Those are the characters. You can see that this is the literature, this is our sample, all same. And taxonomic characters of adult female. Female is differ to male, and front brownish gray lines that are usually indistinct because male has distinct characters. And also orbicular spots elongate, narrowly outlined in black with the pale rim around the dark center. Is transferred white with partial narrow black border. Veins often dark distally. And wingspan is 25 to 35 millimeter. And prepared on maize under local laboratory conditions. Incubation period range was 2 to 3 days, mean 2.3 to plus or minus 4.5 standard deviation. And also larval period, mean 15.40 and 14 to 19 range. And total life cycle from egg to adult, male 30 to 43 days, and female will little longer, 35 to 40. And also, now I am going to discuss about the reproductive biology of fall armyworm under local conditions. Pre-oviposition period is range 3 to 4 days. Oviposition period 2 to 3 days. Post-oviposition period 4 to 5 days. And fecundity 135 to 1,167 eggs at the laboratory conditions. Egg hatchability percentage is 95 to 98%. Male adult longevity is 7 to 9 days, and female adult long. And also, these are the larval characters, the larval growth stages and head capsule width under local environment conditions. And we received, normally, fall armyworm has 6 instar stages. This is the range, 2 to 3 days. First instar, second 2 days, fourth instar 2 days, and fifth 2 to 3 days, 4 to 6 days. And also, this is the head capsule width during first moult. This is the range, 0.3 to 0.35, 3.2 to 0.35, second moult, 0.47 to 0.52, and third moult, 0.4, 1.17 to 1.3, and fifth, 1.87 to 2.05. These are the pictures we received from the lab. First instar, second, third, fourth, fifth, and sixth. And this is the larval laboratory condition. Egg stage 2 to 3 days, larval instars 1 to 6 days, larval stage that because 1 to 6 instars last 14 to 19 days, 9 to 12 days, pupal stage, and adult stage 9 to 12 days. It can be concluded that based on the larval and adult morphological and taxonomical characters, occurrence of fall armyworm in Sri Lanka was confirmed. Further confirmation using molecular technology may be carried out for reconfirmation. Actually, CABI has done, CABI has completed an incubation period of X ranges 2 to 3 days, larval period 14 to 19 days, pupal periods 9 to 12 days. Total life cycle 30 to 43 days male and 35 to 46 days female. And I would like to acknowledge actually Assistant Director of Agriculture, Ms. Madhusha Rajapaksha, AI Mahayoya, she informed them, and others who contributed. These are the references. Thank you very much for your kind attention.
Thank you, Dr. Perra. Presentation is open for comments, discussion, questions. I just like to mention, Dr. Perra, in one of your slides, I saw the length of the larva is about 30 to 35 centimeters. I think it has to be a type error, it has to be millimeters.
Yeah.
Yeah, with this open for...
Thank you. I just want one question from here. Just want to know, is it possible for this pest to be transmitted with imported seeds?
Actually,
I mean, into Sri Lanka?
We didn't we got any information about importation through seeds, but I don't know. But there are so many repercussions. But we have to investigate that when they arrive to Sri Lanka.
Is it possible? I mean, have you? Would you like to comment on whether you are going to do some tests to show that there's a possibility of introducing this kind of pest in the, at least as a prevention for the future? Not only armyworm, maybe other things.
Actually, NPK West, I think Mrs. Hevage and others have done some awareness creation programs to border seaport and airport officers, and also to the necessary stakeholders. Also, therefore, when during importation, and also I think there will not be any introduction from seeds. We have to have strong quarantine procedures to stop as well as other armyworms or other pests, because these days we have another problem at Gaul as well as in Ranwell area. We have to still identify the caterpillar.
And on this transmission to seeds, this kind of pest, with your experience, can you please comment on this? Yeah, please.
With importation of dried seed, very low possibility to introduce into the country. But when we get alert from the APPC, we have improved our plant import permit condition to do the fumigation before sending the consignment to the country. At the country of origin, they should have fumigation the consignment. Inspection for imported maize also. But very few possibility, because if it is contaminated with the pupal cases, the only chance. But normally they are exporting the grains from the silos, and they have stored long period in the silos, and very low possibility to introduce with the
and also the silos temperature is high, therefore the possibility is low.
I have one question. Now it is 1 year since we had this epidemic last year, almost 1 year. Do we have any idea about underlying reasons why we had this epidemic? After so many years, this is the first time epidemic we had in this pest in Sri Lanka. And any underlying reason that you can think why this came, because of environmental or any other reasons? And are we ready? At this age, this time of the year, now it is time in those areas, even the other areas of the country, as a senior officer in the plant protection, are we keeping an eye on those areas to see that we are ready to tackle the problem when it is?
Actually, I earlier described about its movement, 100-kilometer movement with wind. And it was introduced to, I think, May to India. It is easy to come from wind. That is one reason. And also, another thing is that regarding your first question, regarding the second one, actually not me, strong extension service and also plant protection service. My officers contribute a lot to do awareness creation and other activities, and also our whole Department of Agriculture, and also Mahailup Palama, Mrs. Nishanti has done a lot of work to got together and developed IPM package, and also we others are doing some biological control research. I am also doing some research, and I have identified, explored 3 biological agents for the purpose. And I think collaboration and strong links this year we didn't get much problem with fall armyworm.
Okay. Yeah, sir.
Yeah, thank you very much for telling that the adult fly during the night 100 kilometers or whatever you said, right? So because with the wings, right? You have to underline that one, because this was debated in the parliament. How can this that's very important, right? And the thing is, related to the question asked by the chairman, the Dr. Nimalisa Nayak. Now we have a pest. I can't remember the zoological name, but anyway, Godavella.
Yes.
And anatomically, I can see some similarities. So that appears suddenly and spreads all over. Just like, the buffaloes or something like that, so you can see only the root system or the bottom of the plant. So just like harvested, seedlings, right? I mean, with a sickle or something. And this, we never control. But maybe, it appears suddenly, right? Nobody knows. So this was, can be climatic or whatever it is. So when I saw this appearance there, then I told DG, actually, you don't have to worry, it may not appear again, right? So I may be experiencing it again. So during my lifetime in Department of Agriculture, that 31 years, I saw only once this insect, and that was devastating. So similarly, I think there's some sort of, biological agents to control this one. So have you done any study of the biological agents? So that was the question I have. Thank you.
Sir, thank you for your question, sir. Actually, that is Spodoptera mauritius and Godavella, and also Spodoptera litura and Mithymna separata, both three pests occur. Mrs. Nishanti and team, and also the Plant Protection Service also with the collaboration of RRI have done some work. After that, actually for that we had to spray insecticides, which was recommended for recommendation for came from the Spodoptera species. That is Spodoptera. We use tentative recommendation to control fall armyworm, which was used for Godavella, that is Spodoptera mauritius. Some biological agents. And also I think Dr. Hodi team, also Dr. Bandara and others also doing some work on biological agents. And also I found some Cortesia plutellae, not Cortesia, Cortesia species and Trichoderma species to control Spodoptera frugiperda, that is fall armyworm. For fall armyworm, if you are doing any biological agents, as our madams told, we have to do a lot of work and also do mass rearing techniques. At the moment, I think most of us, I am also doing some work, lot of work, and I think in the near future we will have successful research outcome to control fall armyworm biologically.
Dr. Jayan, I would like to ask one question. Can you definitely fall armyworm is eradicated from this island?
Actually, it is little bit difficult, because if we identify from the beginning, we could have done. And also, but another thing is, in any countries, they couldn't eradicate fall armyworm. We, from the beginning, I also thought that we could have eradicate the fall armyworm, but because of their nature, that is 1,000 eggs per female and distribution, will be a problem for, will be an advantage for fall armyworm to distribute throughout the country. Therefore,
that means there's a possibility of building up of this armyworm.
Now also maybe next, during this Maha, sometimes there will be another outbreak, but our extension service and our officers have done a lot of work not to aware the farmers infestation again, because we have a very good IPM package and things like that. And also we are, I couldn't tell, they are practicing pheromones and also some microbes.
I think we have to develop a contingency plan for the control of polymer.
Thank you, Dr. Dani Perra. Now we have come to the end of the time you allocated for your presentation. For her presentation, all of us give her a round of applause. And we go to the next presentation. Next title of the next presentation is Biology and Management of Harpedona marginata, a pest of Dioscorea species in Sri Lanka. Will be presented by, it's not indicated, is it can you please identify yourself, because there are so many researchers in this one.
Yeah.
Who is present in this?
I'm PH Ranavir.
PH Ranavir, right. Thank you.
Chairman sir, judge panel and ladies and gentlemen, I'm going to present biology and management of Harpedona marginata distant, a pest of Dioscorea species in Sri Lanka. This was a BDWG problem in Sabaragamu province in 2016 and 17. Let's move on to the introduction. Yam is a tropical tuber crop cultivated for food and medicinal purpose. Dioscorea genus consists of 630 identified species. Accessions found in Sri Lanka, like popular variety Dioscorea alata, pure Dioscorea esculenta and Dioscorea bulbipera. These are grown commercially in Sabaragamu and Central Province in Sri Lanka, and there was no serious pest death of Harpedona marginata vegetation reported with severe leaf discoloration. And infested leaves are highly prone to leaf diseases. Even death of infested seedlings have been reported. This pest was reported in Thailand. In Taiwan, in Dioscorea field, major pest is the Harpedona marginata. Even though this pest is spread in some countries, there is no well-documented information. Therefore, our objective was to identify mountains and the control measures. Let's move on to the material and methods. Sample collection and pest identification. We collected samples from Hodi Ganora Research Field, Kundasala, Manikina, Kegoli, and some samples were received from the farmers. Pest identified using the key to genera tribe Ictitarsasuni and the voucher specimen kept in Hodi. For the confirmation of the damage symptoms, the healthy leaves were after that symptom developed, leaves were compared with the already received samples. Accession is screening under natural infestation or in the field condition. 33 accessions of edible Dioscorea. Incidences were observed June to September in 2017 and 18, but this screening was done in 2017. Top 50 leaves of each vine were observed and number of leaves with infestation was recorded. Each accession per replicate. We conduct bioefficacy tests under the laboratory conditions. For that, unimpested clean leaves were dipped in insecticide solution for 5 minutes, and those leaves were dried in air or exposed to the Leaves treated with water and dried for 3 minutes were used as the control. For the spraying method, we just spray insecticide solution at once and follow same method. All the treatments were repeated 3 times. Randomly selected same age 5 adults were introduced into the experimental unit. Insect counts were obtained until 7 days. It means live count and the dead counts. Percentage corrected mortality was obtained using Abbott formula. Neem formulation and neem seed water extract and some synthetic insecticide were used in bioassay. This figure shows how we arrange the bioassay in the laboratory conditions. This and the test. And to keep the freshness of the leaves, we introduce smaller cup filled with water, and net materials facilitate the air circulation. For the peel screening, we selected thiamethoxam and neem formulation. 3 replicates per the treatment, and candidate insecticide was sprayed 2 times for the 4 months old Rajala. Harpedona marginata adults and neems were counted just before and after 3, 7 and 21 days. Insect counts obtained top 50 leaves of the vine. Data were analyzed using SAS, pre-counts taken as a covariate. For the life cycle study, sexually matured male and female 1 to 100 unit. And we have to conduct a number of preliminary trials to observe where they lay eggs and how many days taken to lay eggs. For that, leaf petioles and leaf veins were checked daily for the presence of eggs after the 7 degrees Celsius. After that, experimental units were observed for the number of days taken to nymphal stage development, and those nymphs were fed with newly leaf until growing the adulthood. Days taken to emergence of adult was recorded, and the experimental unit were we observed that this fed on lower side of the leaves. Pest damage symptoms on leaves produced in the laboratory and the field were similar. As symptoms, white color patches on the upper surface of the leaves were observed, but the patches turned brownish color with the time. This figure shows how we develop the symptoms develop in the laboratory condition. This is the experimental experimental units, and the symptoms. This figure shows the dorsal view of adult and the ventral view of the male. Male and female both were similar, but female insect somewhat lengthier than the male insect. Male insect 3.5 to 3.8 millimeter in length, is translucent with puscus. Ventral part of the body was dark puscus. Genitalia were pygophore with long, curved and spine-like projection. Female is about 3.4 to 4 millimeter in. This was screening of Dioscorea accessions for this pest in infestation under field conditions. We screened 33 accessions, and all were susceptible for this pest. Higher infested leaves were observed in Dioscorea palata. Damage symptoms vary 12.5 to 90%. The least damage was observed in Guruala and Eshuma. Eshuma is a African accession. This letters are for Dioscorea esculenta, Dioscorea bulbipera, and Dioscorea rotundata. This accession, the species belongs these accessions. Bioefficacy of insecticide, percentage of corrected mortality of this pest after application of different neem formulations. We can see that 80% neem formulation was not effective to control this pest, but 0.75 in EC neem formulation at pest effectively, and neem seed water extracts also was able to control this pest. This table shows the percentage of different synthetic insecticide. We can see that 3 days after application, 100% mortality was observed from thimethoxam applied treatment and imidacloprid and thimethoxam plus chloran traniliflor. This table shows the field screening of selected insecticide for this pest in 2017. We can see that first 2 treatments were able to effectively control this pest even after 21 This table also showed same results. These first 2 treatments were able to control this pest effectively even after 21 days after application. From the life cycle study, we observed that they lay eggs in leaf petiole and the leaf veins. When they deposited eggs in leaf vein and the leaf petiole, the shape of the eggs is oval shape and single egg deposited at a time, not clustered eggs. And to complete the life cycle of this pest, 26 to 27 days taken at 20. This table shows the average number of days taken to complete the life cycle. We can see that higher number of days was observed for to complete the nymphal stage. Conclusions. Harpedona marginata distance feed on leaf sap of the Dioscorea species, leaving white color patches on leaves. Pests observed in June to September in 2017 and 18. All the accessions were tested were susceptible. Harpedona marginata takes 26 to 27 days to complete the life cycle at 27 degrees Celsius room temperature. We should not worry because even at this level of the pest, even a neem formulation. This is the acknowledgement and the reference. With that, I conclude my presentations. Thank you for listening.
We are running behind time. Any comment, question, please? I have just one comment. I saw the yield data and the infection, maybe disease. That was 60% damage. We got some 43 tons per hectare. So I mean, heavy yield.
Yes, sir. This is.
Yeah, this one. Yeah, with the.
Yeah. This is one replicated data. According to this data, sometime with the 90% infested leaves, we observed we get 40 tons per hectare yield, but 87% with second one we got 20 per hectare. This is data because at that time these accessions were cultivated for characterization purpose. So we got that problem, and we want to search some information. That's why we conduct. So with that one replicated data, it's difficult to correlate damage symptoms and the yield. Yeah.
Thank you. Any other comments? So, in the absence of any other comments, questions, shall we appreciate the session? The title of the final presentation is Identification of the Causal Pathogens of Bacterial Wilt Disease in Cucurbitaceae crops and beans, and its alternative wheat hosts in Sri Lanka. This is
Thank you, Chairman, sir. Good afternoon, ladies and gentlemen. My presentation on bean and its alternative wheat host in Sri Lanka. Coming to the introduction, bacterial wilt caused by Ralstonia solanacearum. It's ranked second most important bacterial pathogen species. Its earlier classification of Ralstonia solanacearum according to Racer's BioVars. This is one of the main constraints in solanaceous crops, as crops. In recent years in Sri Lanka, outbreak of bacterial wilt in exotic hybrid cultivation in many places, mainly the central province and east, north western province. And gourd from Marasana, wind bean and bush bean. Here you can see the wilt symptoms, suddenly drooping leaves, whole plant wilt, and leaves become brown and necrotic. Here and beans. When we come into the detection and identification of bacterium, we can use conventional method, initially field symptoms and oosin test, pathogenicity test, virulent test, biochemical and HR test. We can use 16 DNA sequences using PCR with specific primers. So therefore, it is need to accurate identification of causal pathogen of bacterial wilt of cucurbits and bean. When we come into the management, it's colonized in both host and non-host plants, including many weeds without producing visible symptoms. And also in Sri Lanka farmers' fields, continuous cultivation with susceptible host crops in wilt-infected fields, and of Ralstonia solanacearum. Therefore, objectives of the study were to isolate and molecular identification of causal pathogen of bacterial wilt of cucurbitaceous crops and pole bean, the weed host that harbor the bacterial wilt pathogen. Coming to the material and methods. First, we collect the wilted plants and in selected reported incidents and isolation of Ralstonia solanacearum is followed standard techniques. First, we remove the root system, wash the root system to remove soils and bacterial streak on SPA, and pure cultures streak on TZC medium. For asymptomatic weeds, there is no oosin out, so we macerate the basal stem in phosphate buffer and streak. Biodetermination based on their ability to utilize selected disaccharides and hexose chemicals using Hayward's basal medium. And here we use 33 Ralstonia isolates and 25 only characters on TZC medium and observe the change in Haver's medium color green to yellow. And hypersensitive reaction, the bacterial suspension infiltrate on the underside and compare with the recorded data with traces identification. Then we have done molecular confirmation of Ralstonia solanacearum, extraction of genomic DNA from 33 isolates, and PCR and sequencing, PCR products we sent to sequencing in Science Faculty Biotechnology Department and Homology Research. Coming to the results and discussion, here you can see cucurbits and bean produce typical colony characters, creamy white fluidal colonies with pinkish scent on TZC medium after 48 hours. And here you can see characters. Here I show you the some isolates only, and typical colony characters absent on TZC in 3 wheat samples, isolates. Kura Tampala, Nidikumba and Gata Tumba is free. You can see the 25 weed species positive in TZC medium. 3 species is unknown. 22 species belongs to 11 families with infection. The most weeds in our vegetable fields, Hulantala, rubber, Kalanduru, Girapala, Kadupahara and Balathana. This is also reported in other countries. And biochemical test for alcohol by changing the color of the medium green to yellow within 14 days. When we compare to the Hayward Biowa classification, it gives belongs to Biowa 3. And when infiltration, the symptom induce yellowing of the infiltrated area 8 hours after infiltration. According to the races classification, these isolates belongs to race 3. And coming to the molecular identification, PCR product 400 BP with specific primers confirm that all isolates of cucurbits and pole bean belongs to Ralstonia solanacearum. And the identification of isolates of Ralstonia solanacearum in species primers confirm all the Ralstonia isolates, wheat isolates belongs to Ralstonia solanacearum. When we come into the results of DNA sequence, 7 crops and 1 wheat for sequencing, and according to the homology results, it gives highly match within 3 strains, tuberosum in Korea and Cucurbita maxima in China and Solanum lycopersicum in India. And presentation. Bacterial wilting cucurbitaceous crops and Passeolus vulgaris pole bean identified as Ralstonia solanacearum, and all Ralstonia isolates 100% similarity with gene bank and CP note 25986 in China and MF 373815 with accession. And next is all isolates of Ralstonia and 25 out of 28 weeds identified as symptomless carriers of Ralstonia solanacearum. With that, I would say that this finding contributes to update the existing information on development of control measures. And acknowledge Ms. Anuruddhika Abesekara to help me with identification, and Dr. Pradeepa Bandaranayaka, Director AGBC, and Dr. Bhagya.
Now the presentation is open for any comments, questions. We are running behind time, therefore please hurry up. This is the world's second most and so many varieties of cucurbita family and then beans. So it's a very dangerous plant pathogen. So you must have some questions. Until the question comes from the audience, ma'am, now tell me, and once it is infected, it is gone, and there is no way of adding any treatment. So how to avoid getting this disease? That is the most the kind of practical way of
Actually,
getting.
as a pathologist, it's a very big challenge for us. Yeah. Cultivating exotic hybrid varieties. So we received exotic hybrid solanaceous for bacterial resistant. Yeah. Now we are doing. So in future I planning to do exotic varieties seeds and resistant sources. Then we can actually release, not recommend for the susceptible varieties for farmer field cultivation. Now all the cultivated cucurbits varieties are exotic varieties. So we can use some soil amendments, but the problem is farmers practices continuous cultivation in the same land, limited land facilities. So I have observed that biochar is some suppression in control measures.
With agronomic practices such as very well, adding of well decomposed compost or cow dung and things like that, and then having improving drainage, will it improve the resistance of the plants?
It also our farmers cultivation. It's very because colonize in Ralstonia and release the water source. It's spread also water.
Yes, sir.
In cucurbitaceae. That's so.
Pardon,
am I correct? Yeah. So in Japan I had seen, right, so they use bottle gourd seeds.
Bottle gourd.
Yeah. And as the rootstock. So we promoted and provided that bottle gourd is tolerant to bacterial wilt. And there are trainees who had been to Japan, to JICA training, right? They have done this. And find out whether you can do the grafting and solve the problem.
Yes,
just an idea, right? Okay.
Yes, thank you, sir. Actually, solanaceous use in AVRDs in grafting system, but here we not much. Yes, sir. Our varieties. Thinner Western cultivation. Mathili green has some resistance.
But you can screen and see the resistant varieties.
Yes, ma'am. We will continue with varietal screen in our local varieties and exotic varieties as well.
Yeah.
presentation, and we have to congratulate these researchers because this is the first time they recorded this pathogen in Sri Lanka, and so therefore we will appreciate the presentation and research effort with a round of applause. But I'd like to emphasize one more thing again. Dr. Ginadri Soisa, in her presentation, very clearly kind of highlighted the importance of biological control or biological agents, biocontrol. Biological control will work, especially biological organisms will work against bacteria and then bring the population down. So therefore, we need to work towards that. So we need a lot of soil microbiologists, plant pathologists work together. And I congratulate all the technical presenters for their effort taking doing research. And it is not easy, time consuming. You have to have a lot of dedication. I appreciate the findings and presenting here, and especially problems. So information is available, not unlike in nowadays. It's at the touch of a button. There are a lot of information available as what to do to have to get some ideas from the other people. And if you open a smartphone, you get YouTube. Control aphids. That was some rotten rice, cooked rice put in the water. 5 days after sprayed, I did it. I did not see any control yet, but we need to test them scientifically to see whether we need to test them. Please take the time to read this and come up with new innovative ideas of managing plant pathogens and also pests. With this, I wind up this session. I hand over to the coordinator, Mrs. Ilangakorn. Thank you very much.
After the session, I would like to express my gratitude to the chairperson, Dr. Nimal Dissanayake, as well as the panel of judges for being with us and expressing their valuable suggestions and comments, as well as to excellent presentation, as well as the participants for their kind participation. So before that, now I kindly invite Dr. Jayanta Senanayake to present the token of appreciation to the so also remain for a while. I also Dr. Mangalika Nungaliyadda to come forward to take your token of appreciation. University of Sri Lanka to accept your token of appreciation. And finally, Dr. Shanika Jayasekara, Senior Scientist of the CARP, to accept her. Thank you for all for participation of this session, and the lunch will be ready in the Service Training Institute at